Dear students, now I am going to start practical 1, study of mitosis, different stages of mitosis in onion root tip cells. The very first, I am going to explain you the squash preparation of onion root tip for studying mitotic stages. So, you may wonder why we have to study mitosis in onion root tip cells, that is in the plant material. So, the process is same in plant cell and animal cell. The problem with respect to the animal cell, processing of the animal cell, we have to take the bone marrow cells to study the mitosis, or else the tail of tadpoles. So, hurting animals is not allowed, and that is why we have to study this in this onion root tip. So, these onion bulb, they have to be scraped the root region, and then they have to be put it into the conical flask containing water. In 2 to 3 days, you will able to see the growth of root tips. So, these root tips, where you can see there is a highly dividing cells are there in these root tips. So, that is why we prefer these cells material. We have to cut open these root tips after 2 or 3 days the development in the morning hours. In the morning hours means the mitosis will be the the many cells will be in mitosis we will get, that is why. So, morning hours, once we cut the root tips, we have to fix it in the proper fixative. Fixative is the one the solution which stops the postmortem changes. That means we have to arrest the plant material in particular stages or in particular stage of development. That is why we are using fixative. Usually here we are using the fixative Cornoy solution. It is the 1:3 mixture of acetic acid and ethanol. So, for about few hours we have to fix the material. That means we have to keep this cut root tips in the fixative, and then we can transfer it into 70% alcohol. It will remain for a 1 week. We can anytime we can, whenever we are free, we can process it. So, then these whenever we are processing, we have to take out this root tip, take 2 to 3 root tip on a slide, clean slide, and 1 or 2 times wash with the water, so that it is get rid of all the the fixative. Then take 2 to 3 drops of 1N HCl and place this root tip and incubate for about 5 minutes, which loosens the cell wall and helps to penetrate the stain. After this, blot this 1N HCl and put 2 to 3 drops of 2% acetocarmine or acetoarsine stain. So, these are DNA stains. So, chromosomes will stain dark, so that we can able to see clearly under microscope. That is why. So, stain it for about 15 to 20 minutes in 2% acetoarsine stain, and after that you will see the stained root tips like this. So, take only the stained tip portion. So, avoid the rest of the region in a clean slide, and this you have to make it into fine bits with the help of a sharp blade, so that it will spread. Otherwise, it will be like this. So, this if we put cover slip and observe under microscope, it will be like this. So, we can't able to see the stages clearly. So, the cells they have to be spread properly. So, that is why we have to use a sharp blade first make it into fine bits, and with the 1 drop of stain and with 1 drop of 45% acetic acid, now put the cover slip on this, and wrap with the 2 to 3 layers of blotting paper and tap with the rubber headed pencil so that cells will spread now, and then you will it appears under microscope like this. So these in this view hardly you can see any mitotic stages. Many times it will be like that, you can see. So the cells in mitosis is it is a short period, you know, the cells will be in interphase. You have studied already mitosis in your theory. So majority cells they will be interphase, because if the cell cycle is about 24 hours, so 23 hours the cells will be in interphase, in 1 hour they will be in mitosis. Thereby you may imagine how many cells will be in the mitosis. Hardly you will able to see 2 to 3 cells per microscopic field. All these cells are in the interphase stage. During interphase stage you can see the what you see here, there is a rectangular cells on this is a plant cell, and the chromatin material of the nucleus is it is homogeneous and looks granular, and the nucleus has a definite border, that's why they look oval or spherical in shape. So this is how the microscopic the view when you prepare the slide. So how about you see in the prophase? Prophase, I have explained you what all the changes that takes place in the prophase and other things. I am not going to tell you, just I am in the practical part. In the prophase stage there is a coiling and condensation of chromosomes going to take place. That's why chromosomes start appearing under microscope. That is, all these cells are in interphase. You will find this cell. So this is you can able to see, this is in the now in the prophase stage. So if you closer look under in 40x, then you will find like this, you can see. So they start appearing, the chromosomes start appearing under microscope. So in finer view you will able to see like this. So rest of this you can see, these are all interphase. The nucleoplasm is granular, homogeneous. Here you can see the thin thread like structure. So it is during the prophase stage. So at the end of this prophase, chromosome appears a 2 cylindrical structure as a sister chromatids, and nuclear membranes start you can see disintegration, and there is a mitotic apparatus going to form, and all these the condensed chromosome now they align in the equator. That is next stage, it is called metaphase, and you have studied this, they align in the equator. So here how exactly what you prepared slide will be look like this. You can see this is a, so this is, you have to have a keen observation. You can see the alignment of the chromosome in the metaphasic plate, and what all the changes and all you have studied in the theory. I am not going to explain all those. So chromosomes align radially in equatorial region in metaphasic plate. So in the metaphasic plate, the smaller chromosome found in the interior of the metaphasic plate and the larger in the peripheral region. So you will here this is in the plant the centrioles are absent. So the spindle apparatus formed by the spindle fibers, so without a centrosome, or so this is what the difference between the mitosis in plant cell and the animal cell. So the next you will find in the anaphase stage, you will find like this. This is the anaphase stage. So closer look, you will find like this. So during the anaphase stage, the the chromatids they get separate from the chromosome. they start moving towards their respective pole. What you see here, so this is the chromatids movement towards the respective pole. That is what you see in the anaphase. They will take V shape or J shape depending on the position of the centromere. So this is you can see it is a very short period. It shortest stage in the mitosis. It spans about 2 to 4 minutes in onion root tip. So this is anaphase. So the last stage is the telophase. The telophase, already you will see this is the telophase. So they already you can make in closer look, you can see. So they have the chromatids, they have moved towards their respective poles, and the cell is preparing for the division. There is a cytokinesis going to occur. Before the cytokinesis, there will be the cell plate going to form in case of plant cell. So the cell plate going to form. It is not like how you have studied in the animal cell. So this is what you see here. The chromatids, those they have already migrated towards the respective poles. They start reorganizing or decondensation is the process, you can see. So this is what you can see in the telophase. So after telophase, the cell divides and all this process, you know. So identify, this is a quiz for you. Identify the A, B, C and D. Let me know what all these stages from your WhatsApp. Thank you.